Marshes are vital parts of coastlines all around the world, valued for their wide range of ecosystem services. Besides providing habitat for many coastal species and space for recreational activities such as kayaking, coastal marshes also purify water, sequester carbon, and protect human development from damage during storms. In fact, an acre of marsh is valued at over $600,000 for storm protection from flooding and storm surge. These systems are so important. But many coastal marshes are at risk of sea level rise. Marshes are usually able to migrate landward as sea level rises, but as humans build houses and shops and roads, the marshes can't grow landward anymore. This phenomena is called salt marsh squeezing and is a growing problem in Long Island Sound, where 88 to 97% of high marsh is projected to be lost by 2100. One of the strategies managers are starting to consider in Long Island Sound is thin layer placement. Thin layer placement is widely used along the United States Gulf Coast and is basically increasing the elevation of a marsh by adding sediment to the surface. Marshes in Long Island Sound are dominated by a high marsh species called Spartina alterniflora, and this widespread cover of alterniflora is indicative of sea level rise already impacting the marshes, as Spartina typically dominates after loss of other marsh plants due to sea level rise. This is because Spartina alterniflora can tolerate wet conditions. This shift to Spartina occurred between 1940 and 1980 in one of Long Island Sound's largest remaining marsh complexes and continues to happen across the Long Island Sound coastline. Increasing the elevation of the marsh through thin layer placement could allow for other plants to become more abundant.
However, successful restoration requires adapting to the marsh conditions and soil characteristics. A key indicator of thin layer placement success is vegetation growth and recovery through the newly applied sediment, as the application of sediment can either prevent or promote plant growth. Recovery of plant communities is contingent upon the characteristics of the soil. Coastal wetland soils experience varying redox conditions due to tidal characteristics and fluctuations. These changing conditions create the potential for the development of acid sulfate soils, which have a low pH and increased sulfide content.
The low pH comes from the formation of sulfuric acid in the soil when there is an abundance of sulfur. The sulfur will react with oxygen that is introduced as the soil becomes oxidized. Oxidation happens as the tide goes out and water leaves the marsh, letting the soil dry out. Since marshes are tidally influenced, they constantly go through this cycle of drying out and becoming oxidized, then getting flooded and becoming reduced. These variable redox conditions are important for the microbes that act as catalysts in many soil reactions and are responsible for turning soil sulfur into hydrogen sulfide, which gives marshes their well-known rotten egg smell. The variable redox conditions are also a key part of the formation of acid sulfate soils, as oxidation allows the soil to dry out, causing sulfuric acid to form. Acid sulfate soils are common around the world and are problematic in marsh restoration because they are harmful to plants, and successful restoration means that plants regrow in the marsh. Since marsh restoration projects happen on the coastlines, there are a number of ways that acid sulfate soils can be introduced. The first is that the original marsh can either be an acid sulfate soil or potentially acid sulfate, or the second, the material added to the marsh surface in restoration could be sulfitic. Dredge material is often used in thin layer placement as it is readily available in coastal systems. However, dredge is sourced from marine environments where it accumulates sulfides and has the potential to become an acid sulfate soil once it is exposed on the surface. Therefore, amendments could be added prior to deposition on the marsh to counteract the development of acid sulfate soils, increasing and maintaining soil pH, and creating optimal soil conditions for recovery of the plant community. So how do these amendments work? Soil amendments are already used in agricultural settings, altering the soil chemistry to promote plant growth and increase crop yields. To increase the pH of an acidic soil, agricultural lime is used. Therefore, it can also be used to increase the pH of an acid sulfate soil. The primary active ingredient in lime is calcium carbonate. Calcium carbonate is a base and can combine with sulfuric acid to create gypsum, water, and carbon dioxide. This reaction neutralizes the effect of the acid and prevents the pH from dropping. This is why materials with calcium carbonate, such as agricultural lime or crushed shells, can be used as amendments in thin layer placement. Amendments containing iron can also be used, and the same tidal fluctuations that can allow for the creation of acid sulfate soils due to changes between wet and reduced versus dry and oxidized soil can create the right conditions for iron to form solids with sulfur, preventing it from becoming sulfuric acid. Iron oxide binds to the sulfur in soil before the sulfuric acid has a chance to form, preventing the pH from dropping. First, iron oxide binds with sulfur to create sulfur dioxide and iron, which then combine to create iron sulfite. The Landscape Biogeochemistry Lab at the University of Connecticut is conducting an experiment that tests different amendments in coastal wetland soils. These are soil cores, and in each is a layer of coastal marsh topped by a layer of dredge material to simulate thin layer placement. The dredge material has been mixed with different types of amendments of iron oxide and calcium carbonate to prevent the soil from becoming an acid sulfate soil. Pore water measurements are taken weekly through lysimeters placed in each layer. These measurements allow the researchers to understand the chemical processes happening in the soil over time. A lysimeter is a thin tube with pores that gets inserted into the soil to take the pore water measurement. A syringe is used to sample the water in the soil, sucking the water out with the lysimeter acting like a straw. These water samples are then analyzed for different characteristics, such as pH, iron, and sulfide content. The pH samples are analyzed using a benchtop pH meter such as this. Here, the probe on the end causes an electrochemical reaction with the water, and the reaction gives us information about pH. The iron and sulfide water samples are analyzed on this Thermo Fisher Gallery Plus instrument. The water samples go in these racks, and needles inside the machine mix it with different reagents to cause a reaction. The color of the reaction is read by a special sensor and identifies the amount of iron or sulfide in the sample. The experiment is ongoing, but the goal of the project is to provide information on potential amendments that could be used by coastal marsh managers on Long Island Sound as they design marsh restoration projects. Development of thin layer placement projects is increasingly important as new projections show that the sea level in Long Island Sound will increase by 50 centimeters by 2050, more than doubling the historical rise of 4.6 millimeters per year. As managers are developing thin layer placement projects, they will start to incorporate amendments containing calcium carbonate or iron oxide so that acid sulfate soils do not develop and the restoration is successful.